All right, thank you for that introduction. First, apologies from Peter. He wasn't able to join us today, so I'll be tackling this presentation. So thanks everyone for joining. We'll be shifting gears a little bit for this presentation. This is not a particularly scientific talk, but what I'm hoping to do is present some ideas and hopefully get your feedback on how we can really build a cancer community, both locally and within GA4GH. Slides aren't moving. There we go. Okay. So the Global Alliance for Genomics and Health is an international non-for-profit that develops standards, policies, and frameworks for the responsible and secure sharing of genomic and other health-related data. So we aim to enable sharing of data not just across national or institutional borders, but also across the translational continuum between research and healthcare. And so we encourage technology-enabled federated approaches, meaning that analysis is moved to the data rather than, say, aggregated around the researcher. And we do all of these things by convening stakeholders, catalyzing sharing of data, and fostering innovation. And so just to clarify, we do not generate or store any data, and we do not perform research or care for patients. So the GA4GH ecosystem consists of 600 organizational members covering 90 countries. So that means that over 600 organizations have committed to GA4GH's shared mission and principles of responsible data sharing. And while the majority of our members come from the life science technology industry, a lot of the core contributors, so these volunteers that work on developing our standards, come from research institutions and academic medical centers. So how do we work? We're set up kind of like a matrix. So starting on the left-hand side, we have 6 technical work streams and 2 foundational work streams. So these technical work streams, which are focused in the areas of data discovery, large-scale genomics, so that would be things like genomics file formats, data use, cloud, genomic knowledge, which is basically variant representation and annotation, clinical and phenotypic data capture. Yeah, so those are all 6 of them, and all of these streams are the key production teams that build our standards. And then the foundational work streams will provide guidance to these technical work streams. So, every approved GA4GH standard has to go through a regulatory and ethics review. But they also independently develop policies and frameworks that cut across the organization and also have the potential to shape the field a little more broadly. And then these check marks in the middle represent our contributors. And so many of these contributors come from our driver projects, which are these real world initiatives like Genomics England or like ICGC Argo that guide standards development and pilot our tools. But you don't have to be from a driver project to contribute. We have kind of quite a broad contributor base, and as long as you have an internet connection, you can basically join one of our development calls. So these are our driver projects. We have 23 to 24 driver projects. There's a slight difference in number there. You'll see NCI is shown on here as a single logo, but they've actually split into 2, one for the GDC and one for the CRDC. And so these driver projects span different focus areas, some covering more than one. And as GA4GH grows, it's not sustainable to keep adding driver projects. They come with a certain, set of responsibilities, and they make up our steering committee. So we might actually never do a call for driver projects again, but instead refocus around strategic partnerships and groupings of like-minded initiatives, which is basically the basis for this talk today. And so we did launch our first focused community this year, and I'll introduce that shortly. But what I'm hoping everyone here can do today is think about what a cancer-focused community would look like to you, and who or what initiatives should be a part of it. So looking at our driver projects, we do have a solid foundation in cancer. We've got 5 driver projects that specifically focus on cancer, and then we have many others that have cancer within their mandate. And then looking a little more locally, almost all of our cancer-focused projects either are based in or have a very strong connection to Canada. And so there's opportunities here to, leverage these partnerships and really build this collaborative ecosystem. So I mentioned that we launched our first focused community this year. So for several years, in partnership with Genomics England and Australian Genomics, GA4GH has hosted national initiative meetings alongside our in-person meetings. And so these meetings are aimed at bringing together national initiatives around the globe that are focused on advancing a genomic strategy across a country to discuss common challenges and share solutions. And so as this group kind of evolved, we realized a few things. So one, national is a little bit restrictive. There are many genomics initiatives that expand across a region or across a consortium of countries. And two, as GA4GH grows, having this sort of focused collective is beneficial to both standards development, for understanding and fitting the needs of a specific community, but also to initiatives who want a place where they can, discuss real world implementations and systems for, for the implementation of clinical genomics. And so the Genomics and Health Implementation Forum, or the GIF, as it's called for short, is the formalization of those national initiatives to date. And so its main goal is to empower knowledge exchange and collaboration and reduce the duplication of efforts among global genomics initiatives as they work towards implementing genomics in the clinic. And so a lot of the topics that are discussed among these initiatives are driven by the common challenges they've identified among themselves. For example, many of the initiatives have said, I'm having trouble harmonizing clinical data sets. And so they shared various approaches that they all took and sort of ended up coming to the ClinPheno work stream with a specific standards need. And so we held the launch of this meeting in March, and we'll be holding a meeting in association with our GA4GH plenary in a few weeks. And so with the formalization of GIF, we also formalized what it means to be a member. And so there's a few criteria that needs to be met. So for example, initiatives have to be an organizational member of GA4GH. And they also have to be able to demonstrate that they are working towards or supporting the clinical implementation of genomics. And so over the past few months, we've added 16 members, which are shown here on this page. So we're really starting to get together this community of initiatives that can share expertise and build solutions. And so one of the things we heard from a lot of our driver projects is having a standard is one thing, but implementing it is a whole different ballgame. So, how can this standard fit within my system, or how will it interoperate with other standards? And so shown here is the implementation of GA4GH standards in the Australian Genomics acute care genomic test ordering system. And so as an example, they implemented aspects of one of our standards called Phenopackets to record phenotypic features using a standardized format that's shown here on the left. And then they also implemented the data use ontology, which allows you to semantically tag genomic data sets with usage restrictions based on the information provided in the consent section. And then not shown here, they also have a holding place for our Pedigree standard to capture family health history information. And so this is the type of thing that was discussed at a GIF meeting. And what I hope it demonstrates here is that, this type of topic specific presentation and, what other initiatives are doing similar things, is something that initiatives can get together on and learn from and possibly adapt. And so this slide is just kind of summarizing all of these examples. So what can a cancer-focused community accomplish, and how can they, help advance data sharing in oncology? So one, this community can help inform GA4GH on common needs for cancer use cases. They can collaborate on developing implementations and shared workflows and systems. So, discussing how can we build a system based on the building blocks we have available. Who's implemented the different APIs, what data sets are available for us to share with each other. And third, to share resources and expertise. So kind of like the acute care ordering system example, asking questions like, how did that system actually get pieced together? And then what didn't work when you tried to implement things? So that's really valuable knowledge to know as well. So we have this vision for this cancer-focused community and the role it could possibly serve. But I'd like to highlight some of the things we're already doing around building a cancer community. And so this is just showing all of our approved standards, and I'm not going to like dive deep into any one of these today, but instead highlight two that are really driving cancer-specific use cases. So first up is Phenopackets. And so Phenopackets is a uniform machine-readable schema for exchanging both high-level and deep phenotypic information. And so while great strides have been made in exchange formats for sequence and variation data, you know, for example, VCF or variant call format, complementary standards for phenotypes and environment, we don't have it all. So the Phenopacket standard is meant to sort of fit that sweet spot between something simple and something highly expressive, something that's easy to use, easy to import and export, and, you know, easy to comprehend. So this is the basic structure of a Phenopacket. So you've got, you know, the patient up on the left-hand side, the sex, phenotypes associated with that patient, all of which are computed with an ontology. In this example, they've used HPO. So for example, just reading this, they have proptosis, it's severe, and it has a congenital onset. And so this standard was approved in the fall of last year with primarily a rare undiagnosed disease focus. And so we began asking the question, how can we make this more useful for oncology? You know, is there a format similar enough in intent for Phenopackets that we should just be using? Or, you know, what do we need to do to satisfy the needs of the cancer community? And so combining phenotypic data in cancer is a bit of a Wild West. So, you know, there's a number of different formats you can use. The primary one is probably ENCODE, and they've put, you know, a tremendous effort into a high quality and computable schema to describe phenotypic data for cancer, but it's a little more clinically focused, and it doesn't do everything that every project needs. And so this past summer, we've convened a cancer task force, including representatives from ENCODE and all of the initiatives that are listed here, to, you know, sort out what fields need to be added to Phenopackets. So as a starting point, we've been mapping to the ICGC Argo dictionary, so mapping it to Phenopackets to identify fields that maybe Phenopackets doesn't have a good match for, and maybe something that we need to be adding. And then hopefully the end result will be, you know, an interoperable standard useful to a range of cancer projects. And so another one we're working on is the development of our Pedigree standard. And so a number of driver projects have expressed challenges in the representation of family health history information. You know, many groups are still using hand-drawn or non-computable pedigrees, you know, like scan PDF. I think some were saying they were sending pedigrees via PowerPoint. And so there's this really lack of integration with analysis tools. And so there is a format that exists already, and that's called the PED format. And while it works great for a lot of cases and, you know, for basic, you know, mother, father, child trio representation, it doesn't succeed in the representation of more complex families. For example, those involving twins and adoption. It doesn't really have a good way of representing pregnancy status, or even really recording data provenance. So, you know, where did that pedigree come from? So, you know, representation of families is just one piece. So there also needs to be a use for risk analysis, penetrance, and a lot of other calculations that are specifically, you know, important for cancer. And so we've been collecting information from driver projects and tool vendors, you know, on what they need from a pedigree standard. And this is just an example from some of our colleagues at the BRCA Exchange. It's been incredibly helpful. You know, they have particular needs that we hadn't initially considered for inclusion, or, you know, we're still under discussion. So for example, here's a list of some of their required pieces. Age of last follow-up, they deemed as necessary for cancer studies that are essentially survival analyses. Cancer risk can also vary by population. So that would be important to include as well. And then again, just kind of like the Phenopackets example, the idea is to build this standard that can be useful across a range of disease indications. So one of the very exciting efforts we've been working on is looking at how our standards can work in conjunction with each other, and how the implementation of these standards will allow researchers to perform analyses on federated networks of controlled data seamlessly. So this isn't specific to cancer, but is certainly applicable to cancer researchers. And so this is called the Federated Analysis Systems Project, or FASP, and is the central GA4GH forum in which driver project web services and non-driver project are tested for the feasibility of real-world analysis involving multiple systems. So in the GA4GH API ecosystem, researchers can conduct genomic analyses by calling out different GA4GH compliant web services, such as the search API for data discovery. So while searching for data in cancer data sets, for example, a researcher could request phenotype information, say in the form of Phenopackets, in the search results to help identify files pertaining to, say, tumor specimen versus normal specimen, which would then help with knowing which files to submit to the workflow. And then research could also call out passport authorization services, data download APIs like the Data Repository Service, or DRS, and workflow submission and execution APIs, or WES. So for example, maybe they want to pull a differential gene expression analysis workflow from TERS to then run on WES. And so because different APIs can be managed by different institutions, and then they all have different authorization providers, there are many barriers for researchers performing analyses across a federated network. And so the whole idea behind this project is to break down these barriers by aligning the standards and continually performing analysis scenarios and demos using the network. And I really want to emphasize that this is stretching across multiple organizations. So meaning that the researcher can go to multiple data sets at multiple different institutions and potentially do multiple cohort analyses on controlled data. And so it's removing the barrier to doing genomics in the cloud. And so researchers will be able to, execute jobs through somebody else's workflow rather than needing an on-site compute cluster. And if you're interested in learning more about this, we will be having a breakout session on it during our plenary later this month. And so this is just the last project I'm going to highlight. So we're currently working on a survey to determine the number of genome, exomes, and panels we're generating globally, both within cancer and within other disease indications as well. And so this survey is designed to not only inform us of the true pace of genomic data generation, but also validate the need for standards and tools that allow us to capture, analyze, and share vast amounts of data. So if you're generating data, I highly encourage you fill this out. We won't be sharing any individual project data, only aggregated statistics. And hopefully I'll have some really interesting things to share with you in the near future. So we've shared this idea of, a cancer-focused community, an example of a successful so far community, and we've shared examples of cancer-relevant work happening within GA4GH. And so I'd like to, ask you to reflect a little bit and think about some of the key questions. So, what key cancer initiatives should be involved in a community like this? What important cancer-relevant standards should we be aware of? What would you like to get out of a cancer-focused community, and how can we start locally? And so I'd really appreciate your input on these questions with some of the time we have remaining. So I just have a last few slides I'm going to go through here. So key cancer initiatives globally. So I have a listing here. So we've got, the GA4GH cancer-focused driver projects. We've got driver projects with cancer in their mandate, as well as some other key initiatives here. So what are we missing? So I'll just pause a brief second so you can check them all out. and hopefully there's something triggering in your brain, like, oh, there's something missing there. And then what other standards should we be aware of? So, you know, we have a listing of what we see as some of the more cancer relevant GA4GH standards. But of course, there's other really important standards out there that are not within our own remit. So, you know, things like MCODE, like HPO, like NCIT, staging classifications. So again, is there anything here that we seem to be missing? And then lastly, how can we start locally? So, you know, of course, having a strong connection to OICR being our host institute, we also have connections with a lot of other, you know, Canadian initiatives. So really, how can we leverage these groups to build something in Toronto, in Canada, and really help drive, you know, Canadian expertise and innovation? And I believe that's all my slides. So I'm happy to take any questions about anything I've shared. And also I would love to hear your thoughts and ideas on anything we might have missed. So thank you.